Hello student, good morning, and I'm Dr. Manjoo. You are going to study biotechnology with me. So in the chapter we are studying culture of the microbes. So I'm going to discuss about the techniques, that is the genetic engineering techniques. So we have already discussed about the genetic engineering in the chapter number first. So what we are doing in genetic engineering, we are produce the recombinant strain. Here, so how we produce the recombinant strain, we are already discussed in the detail. And here I summarize the genetic engineer techniques here. So recombinant products are formed during the genetic engineering techniques. For example, so I am taking the example of insulin, and the insulin is commercially is called the humulin. Keep it in mind, insulin in market is obtained, or you can search it like as the humulin. It is the genetically modified insulin that is produced by the E. coli, transformed E. coli. So hepatitis B, and that is produced by the surface antigen of hepatitis B. So human growth hormones, interferon, yeah, insulin, hepatitis B surface antigen vaccinations against hepatitis B virus is used. So these are the produced through the recombinant DNA technology or genetic engineering. And I've already told you growth hormones. Yeah, these growth hormones are used for those persons that the growth is retarded. Yeah. So interferons are used in the immunotherapy, and the maximum production of the foreign proteins expression of vector used in such that it replicate to high copy number. So keep it in mind in the simple way, genetic engineering is a technique in which we modify the organism for the large scale production of a particular substance. Or in genetic engineering, we actually modified the genome of a particular organism. So I have already discussed, and different type of products are produced by the genetic engineering, and there is some strains given here such as the Saccharomyces cerevisiae. It's the yeast generally used in the industrial fermentation process. Keep it in mind. And Hestia pasture is the strong inducible promoter. I have already told you. And if you want to produce the large scale production of a protein, so what to do? You add a promoter, that is a strong inducible promoter is added in the before the gene. So that's why in the Fischia pasture strong inducible promoter is added, so by which it is capable of making post translation modification. Where I have already told you, if the prokaryotic gene are expressed in the eukaryotic gene are expressed in the prokaryotic system, so there is a problem of protein modification. For resolving this process in the Pistia pasture, one inducible promoter is added so that the things can be, or you can say the post translation modification can take place. So what we do in the metagenomic, the large number of the microbes genome present in an environment, we are going to isolate these uh, microbes from that environment and study the DNA of these microorganisms. Yeah, so in this case, we are studying the number of DNA, yeah, at the same time of different microbes. That's why it's called the metagenomics. So we are going to discuss the metagenomic in the detail in the next chapter. I hope these points are clear to you.